Hello everyone, welcome to online biology tutorials. In this video, we will be studying how to find amino acid for a given messenger RNA sequence. There is one more video recorded on genetic code. This video along with the video that was posted earlier, this will make your genetic code topic complete. I hope everyone is watching the videos and benefited too. If you haven't subscribed to my channel, please do it. I will be recording the whole book. You will be benefited because you will be getting chapter number 1 to chapter number 16 at one place, at one channel. In case of any query, you may leave your comment in the comment box or on my Instagram ID and Facebook ID. So I'll get back to you within no time. Now for this, it's always better to learn the genetic code table. Especially for the students who are preparing for the competition, they must learn it. And while learning, you need to practice in writing many a times because students, they commit mistake while writing the amino acid. So in this video only, we will discuss what are the various amino acids which are there in the genetic code and how to deduce which amino acid will be there. Okay, so we will take an example and then it will be clear to you. For this, please keep your genetic code table ready. Here I have also taken one for you so that you can easily understand. Now there are various steps. First of all, suppose there is messenger RNA sequence which is given to you. Of course, it will be 5 to 3 prime, right? So let's suppose this sequence is being given to you. And then we will see from the genetic code table how we have to move on. Fine. This is one sequence. Suppose it is being given to you. Of course, it is 5 prime to 3 prime. Now, this is the sequence which is being given to you, and your first job is to find out the initiator codon. So, this green is your initiator codon. So, we will be writing here. This is initiator codon, means the translation process will start from it. Okay. Now we will find out the terminating codon, and here in the table, I'll be explaining through table also because that will make the things clear. See here, UAA, UAG and UGA. These are 3 codons which are not coding for any of the amino acids. So remember, when they will be found in the messenger RNA sequence, they are going to stop the translation process. Okay, so we need to find these 3 bases: UAA, UAG and UGA. Sequence of 3 bases is known as codon. So here we go, and this is our terminating codon. So UGA is our terminating codon, means this will stop the process of translation because transfer RNA will not bring any amino acid from the cytoplasm. Okay, so that means this will be the unit that will be a part of translation. Okay, this will of course stop the process, and the others will bring amino acids. Okay, now prior to this, prior to AUG and beyond terminating codon, means the region which I have colored with yellow. These are the regions which are known as UTR. UTR means these are the regions which will not be translated. So this is the untranslated region of the messenger RNA. So in boards or in any of the competitive exam, they may give you a sequence and they will ask you to find out the UTRs out of it. So it's very easy. First find the initiator codon, terminating codon. The region prior to the initiator codon will be UTR, and the region beyond terminating codon will also not be translated. So it will also fall in the category of UTR, right? Now, your second job is to make the triplets. Okay, so you will make triplets as AUG, CCA, GGC. Okay, these are the three codons which will code for the amino acid. So we will write the amino acid for them. Our job is to find AUG in the genetic code table, and here it is. And it is coding for MET. MET will be methionine. Okay, we will be writing just after completing this every amino acid. Okay, next codon, our second codon is this one. So this one was our first, this one is our second, and this one is our third. So CAA, our next job is to find CAA in the table, and here we go. This is our CAA, and it is coding for glutamine. Okay, so our second amino acid will become glutamine. The third codon is GGC. Now our job is to find GGC, and here I have found for you, and it is coding for glycine. That means with the given messenger RNA sequence, a polypeptide will be formed which will contain 3 amino acids. And of course, whenever you are writing a polypeptide, N terminal has to be written on the left and C terminal has to be written on the right. N terminal means the free amino group of the amino acid. C terminal means the free carboxylic group of the amino acid, which is the last amino acid, right? So the sequence of amino acids in the given polypeptide is methionine, glutamine, glycine. Overall, 3 amino acids are present. So this is the way to solve the problem. Now you know that 20 amino acids are there, okay? So here we will be writing the names of all 20 amino acids that are coming so that you can correlate from the genetic code table. You can see here PHE is there, okay? So PHE means we are talking about phenylalanine, okay? Next is leucine, right? So leucine is the second one. Then here also it is LEU. So LEU means again same. Here the next one is isoleucine. So we will be writing this. We will be covering all 20 amino acids by writing this. Of course, MET, the next one is the initiator codon which starts the process of translation. It's very important, and it is methionine. This we have studied in grade 11, that each amino acid is being abbreviated by a letter, and they can also be abbreviated by their 3 letters that it is seen in the genetic code table. The next is valine, VAL. Okay, you need to learn these in the serial order. So whenever you will start learning the genetic code table, you will speak as phenylalanine, leucine, isoleucine, methionine, valine. Remember, leucine you have to speak twice because it is coming twice. If you will not learn like this, you will commit mistake while writing the genetic code table. So this is the way you can learn the whole table. Learn the amino acids of each column per day. So within 4 days, you will remember the complete genetic code table. Once it is learnt by you, you can practice each day by writing it. But it's very important to learn. We are proceeding to the second column, and the first amino acid given is SER, which stands for serine. Then we have PRO, which stands for proline. Then we have THR, and it stands for threonine. ALA, it means alanine. Okay. Now we are coming to the third column, and the first amino acid is TRY. Okay. This is tyrosine. Then we have stop codons or the terminating codons, which will not code for any of the amino acids. Then we have HIS, which means histidine. Then we have GLN, it means glutamine. Then we have ASN, which means asparagine. By learning the names of the amino acids, it will become very easy because you will retain all the names of the 20 amino acids. Okay, so we are done till here. ASN. The next one is LYS, and it means we are talking about lysine. Then ASP is aspartic acid. Remember, here one thing is again important. Let me pinpoint here ASN, and here I have written ASP, right? So there is a difference. Aspartic acid is a dicarboxylic acid, is a dicarboxylic amino acid rather, whereas asparagine is an amino acid that contains 2 amino groups. Okay, so don't get confused. ASP means aspartic, ASN means asparagine. Right? Now the next one is glutamic acid. So GLU, it is glutamic acid. Again, the important point to be noted here, this is GLN, this is GLU. So GLU is glutamic acid. Again, it's a dicarboxylic amino acid. Glutamine is again diamino group containing amino acid. Don't get confused between GLU and GLN. The next amino acid is cysteine. And we are moving towards the fourth column. Okay. So let me write it for you. CYS, and this is cysteine. The next is the terminating codon. Then we come on to TRP, which is tryptophan. Okay. Next is ARG, and this is arginine. Then serine we have already written. Serine was here also, and serine is here also, so we are not going to repeat it. Arginine also we have written. Here also arginine was there, here also arginine is here, so we will be writing just once. And then we have glycine. So GLY means glycine. We will be now numbering the amino acids. So this was 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and last 20. See here, we wrote at the top that 20 amino acids are found in the cytoplasm, and that's what we have written. I hope this was helping to you. Remember, this topic should be clear before coming to the translation, because the real application of genetic code table comes when we study translation. Stay tuned for more videos. Thank you.